Maple syrup urine disease is an autosomal recessive disease of amino acid metabolism, in which both copies of the enzyme alpha-ketoacid dehydrogenase are deficient. This enzyme catalyzes the degradation of branched-chain amino acids. What three amino acids are branched? Remember that isoleucine, leucine, and valine are the branched amino acids, which you can remember with the mnemonic, I love Vermont maple syrup. Typically, these three amino acids are broken down into alpha-ketoacids, and then further into acetyl-CoA, which can get fed into the TCA cycle. This second step occurs with the help of branched-chain alpha-ketoacid dehydrogenase. Deficiency of this enzyme leads to an increase of alpha-ketoacids in the blood, especially from leucine. Also, a deficiency of thiamine or vitamin B1 will cause an increase in alpha-ketoacids since B1 is used as a cofactor. Why is a branched-chain amino acid disorder termed maple syrup urine disease? Affected infants are normal at birth but develop a characteristic maple syrup smell from their urine. For step 1, if you hear the buzzword maple urine odor or sweet syrup odor, immediately think of MSID and the branched amino acids isoleucine, leucine, and valine. There are also CNS defects such as lethargy, feeding difficulties, coma, and seizures. If untreated, this can cause mental retardation or even death in the first month of life. The treatment of maple syrup urine disease is dietary restriction of all three branched-chain amino acids and supplementation of thiamine, or B1. Alkaptonuria, which is also called ochronosis since it causes cartilage damage and discoloration, is another defect in amino acid metabolism. Here's the larger picture. Tyrosine gets broken down into fumarate through a series of steps. Don't memorize this pathway. I'll focus on the only part that you gotta know for step 1. Just learn the enzyme homogentic acid oxidase, which normally converts homogentic acid to mallyl acetoacetic acid. Alkaptonuria is simply a deficiency in homogentic acid oxidase, which blocks this metabolic reaction. This impairs the degradation of tyrosine to fumarate, leading to a buildup of pigment forming homogentic acids in tissues. What's a major buzzword for alkaptonuria? The most characteristic finding is urine that turns black on standing, which was given the nickname of black urine disease. Other manifestations include dark connective tissue, brown pigmented sclera, and debilitating arthralgia, since the buildup of homogentic acid damages cartilage. Therefore, if you see black urine or black tissue on test day, think alkaptonuria and homogentic acid buildup. Homocystinuria literally means an excess of homocysteine in the urine. This is a disease that shows locus heterogeneity. Do you remember what that is? It's when the same phenotype can be caused by several different mutations at different loci. Don't confuse this with pleiotropy, when you modify one gene and you can have multiple effects in multiple tissues. Homocystinuria has three main causes: a cystathione synthase enzyme deficiency, a decreased affinity of cystathione synthase for pyridoxal phosphate, which is the active form of B6, or a homocysteine methyltransferase deficiency. The treatment depends on the cause. Cysteine is made from cystathione, which in turn is made from homocysteine with the help of cystathione synthase and vitamin B6. Therefore, a cystathione synthase deficiency would lead to decreased cysteine and increased methionine, since the excess homocysteine would be converted to methionine. This latter step will also consume a lot of vitamin B12 and folate. Therefore, to treat cystathione synthase deficiency, you should decrease methionine intake and increase the intakes of cysteine, B12, and folate. In the case of decreased affinity of cystathione synthase for B6, this is best treated by just increasing dietary intake of B6 and cysteine. In the last case, if homocysteine methyltransferase is deficient, methionine becomes essential in the diet. Regardless of the etiology, how do patients with homocystinuria present? Well, they can present with intellectual disability, osteoporosis, tall stature, kyphosis, subluxation of the lens, thrombosis, and atherosclerosis. Here, we see a dislocated or subluxed lens, just in case you haven't seen what it looks like before. Cystinuria is a rare autosomal recessive disorder where there's a defect in the proximal convoluted tubule and interstitial reabsorption of basic amino acids, which include cysteine, ornithine, lysine, and arginine. To remember these amino acids, you can use the memory aid COLA. I like to take this even a step further and think of Coca-Cola, where the first and last letter in cystinuria are CNA, and so is coca. Therefore, cystinuria is the Coca-Cola disease. The defect in cystinuria ultimately leads to the excretion of these 4 amino acids in the urine. One of the most important effects of this disease is that cysteine can precipitate and form kidney stones, which are also called cysteine staghorn calculi. What's the difference between cysteine with an EI and cysteine with just an I? Well, cysteine with an EI is the amino acid, whereas the one with just an I is a dimer of 2 cysteine amino acids that are connected by a disulfide bond. Patients with cysteine kidney stones may have hexagonal-shaped crystals on urinalysis. The urinary cyanide nitroprusside test is diagnostic for cystinuria. To treat cystinuria, patients can be given acetazolamide or citrate to alkalinize the urine. Doing so helps cleave the disulfide bonds in cysteine to form 2 cysteine molecules. It's also important to drink lots of water to stay hydrated. Okay, flash quiz time. This should be an easy one for you. A young boy presents to your clinic with sweet maple odor in his urine. He's diagnosed with an alpha-ketoacid acid dehydrogenase deficiency. What amino acids are not being broken down in this patient? Being the Vermont maple syrup urine connoisseur that you are, you think, I love Vermont maple syrup, representing the amino acids isoleucine, leucine, and valine. Now that we've finished going through the key disorders of amino acid metabolism, let's transition into the regulation of glycogen. Insulin and glucagon are 2 important hormones that regulate the synthesis of glycogen in the fed and unfed states, respectively. Let's go over the mechanism of each. In the fasting state, the synthesis of both glucagon and epinephrine is driven by low blood glucose. Both of these hormones can cause the activation of adenylyl cyclase, also called adenyl cyclase, which synthesizes cyclic AMP, which in turn activates protein kinase A. Do you remember what kinases do? A kinase phosphorylates its substrate, and in this case, protein kinase A phosphorylates the enzyme glycogen phosphorylase kinase, which activates it. Glycogen phosphorylase kinase, which again is a kinase, can then go to phosphorylate the enzyme glycogen phosphorylase, which also activates it. The role of glycogen phosphorylase is to break down glycogen into glucose via a process called glycogenolysis, which increases blood glucose to address the fact that you're in an unfed or hypoglycemic state. Remember that glucagon and epinephrine cause the enzyme glycogen phosphorylase kinase, which can lead to the breakdown of glycogen reserves. On the other hand, what happens in the fed or hyperglycemic state? When blood sugar levels are high, insulin is secreted from the pancreatic beta cells. Insulin activates a receptor tyrosine kinase by causing it to dimerize, which sets off an intracellular signaling cascade that results in the activation of 2 protein phosphatases. Remember what phosphatases do? These dephosphorylate both glycogen phosphorylase kinase and glycogen phosphorylase, causing both to become inactive. Consequently, glycogenolysis does not occur, so glucose remains stored inside cells to prevent hyperglycemia. A less high-yield pathway occurs in muscle, in which calcium and calmodulin can activate glycogen phosphorylase kinase. This coordinates glycogenolysis with muscle activity, ensuring that enough glucose is present to make ATP for muscle function. Here's what it looks like all put together. Sometimes it can get a little confusing if you dive right into this subject with a full picture, so make sure you break it down if you feel overwhelmed. Some general rules are that glucagon and epinephrine result in the phosphorylation of most enzymes tested on step 1, whereas insulin results in the dephosphorylation of these enzymes. These phosphorylated enzymes ultimately lead to glycogen getting broken down to produce energy. We just talked about how glycogenolysis is regulated. Now let's go over what actually happens during both glycogen synthesis and glycogen breakdown. So this is what glucose looks like. It has 6 carbons, which I've labeled 1 through 6 here. Glycogen is a polymer of glucose. Branches of glucose chains are connected by alpha-1,6 bonds. Side linkages, or points of divergence off parent chains, have alpha-1,4 bonds. Seen here is a glycogen chain that is being created. Since we numbered the carbons in our glucose molecule, the 1,6 and 1,4 indicate which carbons are connected. This means that most of the glucose molecules are connected or linked between the 1 and 4 positions, whereas in branches, which we see here, these are connected at the 1 and 6 positions. As you can see, if you keep connecting with 1,4 linkages, you end up with a straight line, whereas the 1,6 linkages come off at a branch point. Why are we talking about these linkages? By storing glycogen in a linear form with multiple branch points, long polymers of glucose can be stored in a more compact form. When glucose levels drop in the body, this stored glycogen can get reconverted back to glucose, which can get fed into glycolysis and then the TCA cycle for ATP production. What's the difference between skeletal muscle and the liver with regards to glycogen? Well, in short, muscles are greedy and keep all of the broken down glycogen for themselves, while the liver is generous and will make glucose for the whole body. In skeletal muscle, glycogen undergoes glycogenolysis to provide glucose, and therefore ATP to be used locally in that muscle during exercise. In the liver, glycogenolysis is not used to increase glucose locally necessarily, but to maintain blood glucose levels, and therefore the energy supply of the whole body. We'll now demonstrate the mechanism of glycogen synthesis. Where have we seen glucose 6-phosphate before? Hopefully you recognize it from glycolysis, where glucose gets converted to glucose 6-phosphate to become trapped inside the cell. First, glucose 6-phosphate is converted to glucose 1-phosphate by the enzyme phosphoglucomutase, which simply moves the phosphate around within the molecule. Glucose 1-phosphate can then be converted to UDP glucose with the help of the enzyme UDP glucose pyrophosphorylase. UDP glucose is a substrate or building block for glycogen synthesis, which synthesizes glycogen by adding UDP glucose to the end of a glycogen chain. Glycogen synthase is activated by glucose 6-phosphate, so if you have a lot of glucose 6-phosphate building blocks lying around, you might as well store it as glycogen. Here we see lots of alpha 1,4 bonds to create a nice long glycogen chain. The body doesn't want just one long inefficient chain of linked glucose molecules, however, so what can the body do to increase storage efficiency? Once the chain has grown to about 10 glucoses long, an enzyme called branching enzyme takes the last few glucoses of a chain and moves them back to the previous 6 carbon on the chain. Adding branch points makes this glycogen chain more efficient to store. If you're following along in our book, you'll notice the figure stores the first 3 main steps that are used to synthesize glycogen. Next, we'll talk about breaking it down. Looking at this pathway, we see that glycogen synthesis steps are located on the right. Since we just covered that, let's take a look at the left, which is involved in glycogen breakdown, or glycogenolysis. Glycogen phosphorylase adds a phosphate group to a glucose molecule at the end of the glycogen chain, separating it from glycogen. Where have we seen this enzyme? This is the same glycogen phosphorylase that we talked about earlier, which regulates glycogenolysis along with the enzyme glycogen phosphorylase kinase. This enzyme can sequentially break down 1,4 linkages in the glycogen chain until they get to a length of about 4 glucoses left. This 4-glucose structure is called a limit dextrin, and glycogen phosphorylase can't break this down any further. To address this, 4-alpha-D-glucanotransferase, part of debranching enzyme, is used to move 3 molecules of glucose 1-phosphate from the branch to the end of the chain. Then, alpha-1,6-glucosidase, which is just the other part of the debranching enzyme, swoops in to cleave off the last molecule of glucose 1-phosphate from the chain. Thanks to debranching enzyme, our chain is now rearranged, which allows glycogen phosphorylase to resume its function of chopping down the long glycogen chain. In a smaller side reaction, glycogen can also be degraded by lysosomes. This requires the enzyme lysosomal alpha-1,4-glucosidase, which is also known as acid maltase. As its name suggests, this can only break apart 1,4 linkages in glycogen, slowly converting glycogen into free glucose. Okay, flash quiz time. What are the end products of glycogenolysis? We just covered how glycogen gets broken down, but the end product of glycogenolysis in the liver is glucose and free phosphate. If you remember from earlier, the muscles are greedy and keep all the glycogen breakdown products for themselves. Therefore, muscles break down glycogen into glucose 6-phosphate, which can then enter glycogenolysis and make some ATP.